Okay, that's good. All right, so I apologize. There's been a little problem with Dr. Emanuele Brai from the University of Piemonte. For some reason, he's not able to get in. So I'm just subbing for him, at least for the first talk. And thank you, Dr. Aniko Korosi. Aniko Korosi will talk about brain food and how food can influence our brain health, and how this subject is so important in preventative measure towards a healthy brain aging. Thank you.
For this invitation, and today I will talk about the effect of nutrition on brain health, but actually with like a focus on how to use this knowledge and what we know so far, and how to improve how we gather knowledge on this topic towards policy, and how to increase the impact of this. So I will be presenting to you about the EBRA, the brain food clusters within the EBRA, and I will explain to you various aspects of it. So first I will touch upon a little bit on what is the evidence that we have for nutrition impacting on brain health, that we miss, what are the mechanistic insights, what are the current challenges with studying the impact of nutrition on brain health, and which of these challenges actually solo efforts but need to be addressed at much higher levels, at the level of the EU, for example, which will lead me then to talk to you about the EBRA, what the EBRA is, and the EBRA brain food cluster is its main activity. So what is the evidence that we have that nutrition impacts on the health? I think all of us here, all around us, all the time, all kinds of recommendations, specific nutrients that can help us think better, age better, have a better functions, Mediterranean diet, supplements, of that kind of supplements. And indeed, there has been a lot of gain in interest in this, but there's also a lot of actually a lot of information missing, and a lot of information is actually not really strongly evidence-based. So I am also a member of the ECMP Nutrition Network. This is chaired by Roger Adan and Susan Dixon. And this is really a network within the Psychiatry Society in Europe, where we try to highlight the relevance of the evidence that is out there on the effects of nutrients and nutrition on brain health, but also what is that we lack and how can we fill. So in this position paper that was written a few years ago, you can see here the complexity of it all, and you can then already start grasping why is it also complex. So clearly, within our clinical assessment, there will be many other aspects next to our dietary exposures and that are determinant of our brain health, such as age or kind of other habits, genetic background, of course, metabolic health. Do we move a lot? Do we exercise a lot? Do we take medications, etc.? And all of this together, of course, makes the questions really complex. And then even further, when we think of mechanisms and in general how to use this either as a biomarker or as a target for intervention, we realize that it is very, very complex. It definitely links our peripheral to our brain, and how the certain nutrients get absorbed and get synthesized and get transferred to the brain. And in addition to that, there are all kinds of characteristics within the brain that will also how all of this is happening. So this makes all of it complex, nonetheless very powerful and very promising. In fact, there has been a really evidence for the association between nutrition and mental health. In this screen, you can see a couple of these papers that are relevant to this topic that have been collected. All the information from various cohort studies that have been done, where nutritional intervention have been done in the context sometimes of Alzheimer's disease, but also of depression and even of anxiety-like disorders, and mostly a lot of them also focusing on simply modulating cognitive functions. Evidence, and this is becoming really undeniable, but there is also a clear number of challenges. So when we think of the challenges that we have at hand right now for bringing this field forward, is that yes, we have this association between nutrition and mental health, but we lack really often causality, and we have quite understanding of the underlying mechanism of how this works. And the diet is often actually not working, of course, on it's combined with lifestyle, and so this has the potential to for prevention and for treatment, and even to modify drug treatment effects. But how specifically, for who, for when, and what exactly, this still remains based on evidence. The data that are out there are informative, but often still limited methodologically. And a number of problems arise because the population is quite heterogeneous, which is true for every kind of population study, of course. But there is also a lack of biomarkers to adequately stratify within and across this heterogeneous population. So, for example, we can envision, if we are thinking of fatty acid supplementation, then of course maybe think of stratify maybe the population based on their original deficits. But this in the existing studies has not been consistent yet. So at this point, really large heterogeneous populations that have been submitted to dietary interventions, but because of this, sometimes these are difficult to evaluate. Most have relatively small sample size still. There's a lack of blinding because of the nature of these interventions often. In addition, of course, we need more insight into the mechanism and understanding exactly how this works at the level of the metabolism, but also the level of within the brain. We need to understand a little bit more about how the genetic background of each individual contributes to the nutrient working or not working for certain specific individuals. And then, last of course, the nutrients differ a lot across the lifespan. And so to provide proper recommendations from improving mental health, we also need to really drive into the knowledge of nutrition. And this is also true of women, so sex-dependent effects as well. Think about, for example, the mechanistic understanding how food impacts on the brain health. There start to emerge more and more papers that try to speculate a lot about what could be the potential mechanism by which this is working. And this is quite a nice paper, but also members of this ECIP nutrition network are involved in. See that indeed is a really large number that are proposed, and this is also what makes it so different compared to a drug, because a nutrient is always going to have a much broader effect. So here some of the nutrients that are being here proposed, for example, are phytochemicals, vitamins and minerals, omega-3 acids and fibers, and we know that in a healthy diet, when there's more of these individuals will have a reduced risk to develop depression and obesity. It's being thought that some of the effects are possibly mediated by neurogenesis, which is the birth of new neurons within the brain, but also the inflammatory system, inflammation central, so in the brain, but also peripherally. Epigenetic modifications seem to be relevant. The HPA axis, so the neuroendocrine stress axis that is being activated. The gut microbiota, of course, is receiving enormous amount of attention, but also the energy providers of within every single cell in our body, the mitochondria. And this is just a number of them. And so I just want to show you, for example, some examples from actually my own research line of how we try to tackle this and how we try to increase our understanding. And we know that early life adversity during critical developmental periods is actually a really big risk factor for a lot of mental health problems later in life, such as depression, but even Alzheimer's disease and dementia, cognitive decline. And we also know that a lot of these disorders are actually comorbid with metabolic derangements as well. So possibly both of these have an early life origin. And when there is an adversity encountered during the sensitive developmental period, we will have an exhibition of this elevated risk, which will be also different for men. So interested in understanding is like what role that nutrients might play in this long-term effects of this early life adversity, and can we use this intervention to protect against this negative early life adversity. So we work in a preclinical mouse model here, and we explored first the fatty acids, for example. So the fatty acids, I think, were about the omega-6 and omega-3 fatty acids, how the omega-3 are more beneficial. And these have really specific, these fatty acids are really important in all kinds of different properties with the brain. Brain development, but also membrane properties, this neurogenesis that was just mentioned, also really important inflammatory modulators. And so what we did is actually expose like our mice to life adversity or just being control, and we expose them to a diet that was either really increasing the availability of these fatty acids or not. And then we looked at later in life, after long being done, the adversity has been done, to see whether we are able to modulate the effects of of this early life adversity with nutrients, with nutritional strategies. And so what is firstly that the early life adversity by itself actually changed the nutrient composition of the brain. So the fatty acid composition of the brain was altered just by this early stress exposure. In addition, what we saw is that when we supplement this diet early in life, which is then basically transferred to the offspring by the mother, because this is during this early life period when the offspring is still breastfeeding, we see that actually this is transferred via the mother, so via the nutritional intake, and it reaches the brain, and we manage to restore the fatty acid composition of the brain in the offspring. And not only that, but also when we look at later in life, we can see that on many different levels, we manage to protect negative effects of the early life adversity. So what you see here is 4 bars. The first 2 are the ones under the non-supplemented diet, and these are the control animals, the stressed animals, and these are the supplemented groups. First the controls and then the supplemented. And what you see here, this is representing a deficit in cognitive functions, which is no longer there when these animals are supplemented. This is a deficit in this neurogenesis that we talked about, which is no longer there in the supplementation. And this is a change in neuroinflammatory cells within the brain, which were increased in the pro-inflammatory state in the brain from the stress exposure, which is also no longer there with this diet. So this is really one of the ways we try to understand how can we use nutritional intervention. We try to test different time windows. Here we are focusing on a really early time window. And then as follow-up studies, of course, we try to really understand even more in detail how this diet does its job, basically. Another interesting nutrient that maybe it's worth mentioning, just to understand how powerful actually nutrients can be in context, are the essential micronutrients. And essential, both in this case and also for the fatty acid I missed to mention, is that they need to be taken in from the diet, so they cannot be synthesized by our body. Essential micronutrients, you have to think of vitamin B6 or B12 or methionine or choline. They are all part of this biochemical cycle, this one-carbon metabolism, which is really important for generation of methyl groups. And these methyl groups are methylation groups that are used for DNA methylation and histone. And these are the basic elements of epigenetic modifications, which I don't have now time to look into, but it's a very, very particular way of modulating gene expression of within the body. Nutrients will determine how effective this kind of cycle is, and that's why there will also depend on the availability of these methyl groups and how well this epigenetic machinery can work. And so we did actually a similar study to the previous one, but instead of fatty acid, this time supplemented with micronutrients. And what we see here again is, first of all, that the stress alone changes nutrient composition of the brain. We saw it this time with methionine, which is one of these essential amino acids. And again, when we supplemented this diet early in life, what we saw is that these were restored, were no longer reduced. And again, we had seen here the cognitive functions being impaired in the standard conditions with this early life adversity, which was no longer the case after the supplementation. And here the mechanism, then we see that actually this seems to be modulated by the HPA axis. I hear some echo.
Yeah, sorry, we have. Sorry, I apologize for that. We have to stop now because it's already 15 minutes over.
But we start, right?
Okay, so you can finish your talk. Yeah. So you can.
We started several minutes later, I think so.
Okay.
Anyway, so this was due to, and that was one of the modulations. So this gives you a nice insight about the mechanistic aspects of this. So what I wanted to mention, then I'm going to skip a few slides because I think I still wanted to get to the really important part, which is the conceptual and methodological challenges with studying the impact of nutrition on brain health is that nutrients rarely act by themselves, often affect multiple processes in the body. The studies are difficult to design and require a large number of participants. And the personalization is also difficult because there are so many individual differences in responses to diet. The dietary assessments and human interventions are very complex. And of course, there is a lot of lack of scientific evidence. So there is a lot of knowledge in the field that is not really well substantiated yet. But is that what needs to be addressed at the EU level is that the involvement of more stakeholders at the level of doctors, patients, and citizens who currently have a relatively poor understanding of what healthy nutrition is. There is indeed a lack of solid evidence to demonstrate that a healthy diet and adequate nutrition impact on the brain, so we need to strengthen that. but also it's really important to realize that foods are not drugs. So it's really difficult to put this in the hand of, for example, a nutritional company, because nutritional supplements are difficult to, for example, patent, and so the turnaround for this is very very difficult to realize. Also from more governmental initiatives. So I just wanted to conclude with saying that this is how this whole idea of the EBRA was. So this is this European Brain Research Area project, which consists of a collaboration between the European Brain Council and some other partners. And within this, we formed a cluster that we call Brainfood. And the cluster is really meant to increase public awareness of nutrition, that nutrition matters from the brain health, and that needs to be, and how to prioritize this at the EU level and at the policy level, and how to manage to then draw more attention to this field.
Okay.
I think the time is up, then I will stop here.
I think we will discuss all of this in the panel discussion, because it's very interesting. And so thank you so much. Bye. I see you later.